All right, today we're diving into something amazing. We're going to be flipping some of the most important switches in all of biology, the secret controls tucked away inside our cells that literally decide who we are. Okay, so let's start with a really simple question. Why is a heart cell so different from a brain cell? I mean, really think about it. One spends its entire life just beating, rhythmically, while the other is firing off these incredibly complex electrochemical signals. They have totally different jobs, right? So why?
Well, here's where it gets wild. They have the exact same DNA. Every single cell in your body, whether it's in your brain or your big toe, has the identical genetic blueprint. So if they're all reading from the same instruction manual, how on earth do they end up with completely different jobs?
The secret, it turns out, isn't in the code itself. It's all about how that code gets read. And that, my friends, brings us to the absolutely fascinating world of epigenetics.
So what is epigenetics? Well, the best way to think about it is like a computer. Your DNA, that's the hardware. It's fixed. It is what it is. Epigenetics, on the other hand, is the software. It's all the programs running on top of that hardware, telling it which parts of the code to use, when to use them, and even how loud to turn them up. Okay, so how does this software actually work? Well, it all comes down to these tiny little molecular switches that can literally flip a gene on or off. One of the most important of these switches is a process called DNA methylation. Now, it sounds complicated, but the idea is pretty simple. The body just sticks a tiny little chemical tag, it's called a methyl group, right onto a gene. And this isn't so much a hard on-off switch, it's more like a dimmer switch. When that tag gets added, it turns the gene's volume way down, basically silencing it. It really is an elegant little process. You've got a gene, it's active, it's doing its thing. Then this special enzyme shows up, slaps a methyl tag on it, and poof, the gene is silenced. That's it. And this is exactly how a heart cell knows it's a heart cell and not, say, a stomach cell that needs to make digestive juices.
And of course, the body isn't just sprinkling these switches around at random. No, they're clustered together in these really important areas called CpG Islands. You can think of these as the master control panels for our genome, sitting right there next to the genes, making all the big decisions.
And just how important are these control panels? Get this, a whopping 70% of all human gene promoters, those are the on switches for our genes, are found inside these CpG islands. That's not a small detail. That's the main operating system for our DNA. So who are the workers doing all this? Let's meet the molecular mechanics. They're a family of enzymes called DNA Methyltransferases, or just DNMTs, to keep it simple. And they work in two distinct teams. First up, you've got the builders. These guys, DNMT3A and 3B, are the architects. They show up early in development and draw up the initial blueprints, deciding which genes get silenced in which cells to create, a liver cell versus a skin cell. Then you have the copier, an enzyme called DNMT1. Its job is simple but crucial. Every single time a cell divides, it makes a perfect copy of that original blueprint. It's the reason a skin cell makes more skin cells forever. What's really cool is that this same basic tool, methylation, is used for totally different things in different forms of life. For us complex eukaryotes, it's what allows us to have all these specialized cells. But for prokaryotes, like bacteria, they use it for something much more primal. They use it as a weapon.
Okay, check out this brilliant strategy. A bacterium covers its own DNA with these methyl tags. It's basically writing "this is me" all over its genome. Why? Because that makes its DNA invisible to its own defense enzymes. So when a virus comes along and injects its DNA, which of course doesn't have these special tags, the bacterium's enzymes immediately spot it as an invader and just shred it. It's a primitive but super effective immune system.
So yeah, this system is absolutely brilliant when it's working right. But what happens when those switches get stuck or flip the wrong way? Well, that's when we start to see the epigenetic roots of disease. You see, in a healthy body, these switches are absolutely vital for controlling cell growth. They're the guardians that make sure cells only divide when they're supposed to. So what happens when that system breaks?
All right, think of it like this. A healthy cell has genes called tumor suppressors. They're literally the brakes on the car, stopping growth from getting out of control. But in cancer, the cell makes a catastrophic mistake. It starts sticking way too many methyl tags on those brake pedal genes. This is called hypermethylation. And what happens? It silences them. The brakes are gone, and the cell just starts growing and growing and growing.
And it's actually a double whammy.
The cancer cell turns off the good genes, those tumor suppressors. But at the exact same time, it does the opposite to the bad genes, the oncogenes that scream go. It removes their methyl tags, turning them on. So you've got the foot slamming down on the accelerator at the same time the brakes have been cut. It is a perfect storm.
But this isn't just a story about what goes wrong, because understanding exactly how it breaks has opened the door to something incredible, a whole new frontier of medicine based on, well, hacking the code to fix it.
Which brings us to a really powerful question. If faulty methylation, these broken switches, can cause disease, can we go in there and fix them? And the answer, amazingly, is yes. The strategy is actually beautifully simple, in a way. We can design drugs that go after the very enzymes that are messing up, those DNMTs we were talking about before.
And this is already happening. It's leading to a revolution. We have drugs called DNMT inhibitors that are being used to fight cancers like leukemia. How? They basically tell the enzymes to stop silencing the good genes, turning those brakes back on. We're even using this idea to develop new antibiotics that mess with the bacteria's methylation, crippling its defenses.
And this is not just some far-off idea. As the researchers in this field say, the potential here is just endless. Our methylome, that's the entire pattern of methylation across all our DNA. It's this huge, unexplored territory for finding brand new ways to fight disease.
So, what's the big picture here? It's that our destiny isn't just locked away in our DNA hardware. It's also in this flexible, dynamic, and potentially editable software running on top. And that leaves us with a huge question, right? What other secrets are hiding in that software, just waiting for us to figure out how to rewrite them for our own health?